it's Talia from Thompson STEM Engagement here, and everybody knows genetics is my jam. And as I'm sure you already know, inside every cell, our DNA holds the information needed to create us. However, the expression of these genes must be regulated by external influences. Similarly to this glow stick, external factors, for instance my fingers, are needed to activate the glow stick. Right, so it's time to talk about epigenetics, guys, but first, roll the intro. The study of epigenetics can broadly be split into two categories: developmental epigenetic events, referring to the regulation of gene expression within embryo development, and environmental and behavioural epigenetics, underpinning the potential for the things we do in our lives to have a heritable effect on our genetic profile. Crazy, right? Epigenetic mechanisms help to add a level of control to the fine-tuned process of gene expression. Without this, we could end up looking like all sorts of crazy creatures, with eyes where our fingers should be, noses in our navels, and gosh, I bear to think. Anyways, although there are other mechanisms at play that are responsible for developmental control of the body plan and fetal formation, epigenetics has a major role in gene expression. And unlike genetic changes, epigenetic changes have the potential to be reversible. As they do not actually change or affect the DNA, it's more acting as like an on-off switch to control genes and their expression and how they get transcribed to become proteins. This is achieved through DNA methylation, histone modifications, and non-coding RNA interactions. DNA methylation works by the addition of methyl groups to particular bases within our DNA, corresponding to the open reading frames, resulting in silencing of that gene expression. Methyl groups can be found in common household items like methylated spirits. A general rule of thumb is methylation events equals turning off, demethylation events equal turning on DNA. So you can kind of imagine DNA methylation as a switch, controlling the expression of particular genes, kind of like the glow stick analogy I showed you earlier. Histone modifications, on the other hand, do not directly affect the DNA. But as the name suggests, the epigenetic changes arise from modifications to the histones. Histones are a family of proteins that function to help condense and supercoil DNA. If you imagine histones kind of like these Velcro balls, and DNA like a piece of string, chemical modifications that take place between individual histone tails binding the histones closer together, and the DNA is bound tightly around these histones. As such, it's unreadable in this compact state. This is referred to as heterochromatin. DNA can only be translated and transcribed into a protein when it's loosely bound around histones, otherwise referred to as euchromatin. Whilst non-coding RNA epigenetically influences gene expression simply by attaching to and silencing coding RNA used to make proteins. So, as we can see, we have evolved to have epigenetic mechanisms for the regulation of gene expression at every step along the way, through modifications in our DNA and histones to RNA, all necessary for the creation of functioning proteins. The crazy thing about epigenetics is these events don't stop taking place in the womb. Oh no, it doesn't stop there. This carries on throughout our lifetime. Studies have shown differences in the epigenetic profile at birth to adulthood, with current evidence suggesting that the highest methylation is seen in newborns and the lowest in the elderly population, perhaps suggesting that epigenetics could hold part of the key to the time-old question of aging. Now that really is an interesting topic. Let's not get carried away though. It's been suggested by scientists that some epigenetic changes required have the ability to be reversed. For example, when looking at smokers, non-smokers and former smokers, smoking results in epigenetic changes to the AHRR gene. The level of methylation to this particular gene can be seen decreased in smokers and significantly so in heavy or long-term smokers. However, once an individual quits smoking, methylation events increase, and with time this can reach a level comparable to the non-smoking population. Some bacteria and viral infections also have the ability to cause epigenetic changes, with bacteria such as tuberculosis resulting in histone modifications, weakening your immune system. Methylation of particular genes can result in things like cancer, as seen with the methylation in the BRCA1 gene, responsible for a type of breast cancer. And if this wasn't crazy enough, epigenetic changes don't just affect us, but they can go on to affect our offspring. During the Dutch hunger winter famine of 1944/45, it has since been observed that children born to mothers that were directly affected by this famine had increased and decreased levels of methylation for particular genes, resulting in increased levels of particular diseases such as heart disease, schizophrenia, and type 2 diabetes. Epigenetics is kind of cool, right? Kind of scary though too. Like, imagine everything that you do has the ability to change and affect your gene expression. That's kind of deep. It makes you think twice about a few things, eh? Epigenetics is super interesting, and it's such a useful tool for the control of gene expression. It's not the only tool though. So let's talk about genetic engineering for real. Broadly speaking, genetic engineering can be split into two fields: somatic cell engineering and germ cell line engineering. There's a wide variety of genetic engineering techniques utilised today, from microbial vectors, transposons, microinjections, non-transgenic molecular manipulation, and one of my favourites, CRISPR-Cas9. CRISPR is an amazing little tool that was discovered in 1987 by Yoshizumi and Nisho. CRISPR-Cas9 works like a pair of scissors, allowing us to remove targeted faulty genes and insert a desired healthy sequence. The Cas9 enzyme associated with CRISPR can be tagged with interchangeable guide RNA, complementary to the targeted DNA sequence to be removed, allowing for the formation of double-stranded breaks in the DNA and the programmed insertion of DNA. And I personally think this is insanely cool. The potential for a genetic engineering tool like this is incredible. Imagine being able to cure any genetic disorder simply by removing a faulty gene and inserting a healthy copy. So really interesting work is going on in this field at the moment, and hopefully this technique can be used in the future to aid in the treatment of particular complex genetic disorders caused by a host of different mutations. But that is a story for another time. Another example of genetic engineering you might be a little bit more familiar with is that we see in our own domestic animals. Domestic animals have been bred by humans for specific traits that we have decided are desirable, whether it's those short little legs on a dachshund or the spots on a Dalmatian. These particular traits have been selected and bred for because we have decided that we like them, and as such we have created artificial selection. For this exact reason, chickens produce double the amount of eggs today as they did in 1950 due to the selective pressure that we put on them. It's funny to think, we still know so little but like to think we know so much about everything, especially when it comes to genetics. But dare I say, this is one of the fastest growing fields in science, and we'll know more tomorrow than we did today. Anyways, I think that's all for today, guys. Thanks for joining me here at Thompson STEM Engagement, and don't forget to like, comment and subscribe and carry on learning.